Okay, I think we're good to go. Hey everyone, welcome to the quarterly Pesticides and Public Health Working Group meeting. The purpose of which is to develop a shared understanding of the public health issues related to pesticide use and to work collaboratively to reduce their application in Iowa. I just threw the notes and agenda document into our chat one more time. Our agenda is pretty strictly co-learning today. We have Dana, Darren, and Greg here to talk to us mostly about neonics. And neonics was a really popular topic for us a couple calls ago, where we discussed what was going on in Meade, Nebraska, that tragedy with the Alton ethanol plant. So we're revisiting what's going on specifically in Iowa with regards to neonics. We will have hopefully a pretty robust discussion about sort of what's been learned about the state of water and neonics in Iowa and what is left to be discovered, what implications there are from the results of these studies that the expertise on our call has done so far. And then I want to have a very brief update on the direction of the working group for you all, as Kamyar and I at the CEEE are thinking about funding opportunities, maybe thinking about how to expand the work of our group to do some action agenda planning development, just to let you know sort of what it is that we're thinking and maybe the direction that this could go. Then we'll do our updates and plugs as usual and close things out hopefully on time. But first, because we have some people who are new on the call and we have some people who haven't joined us for a while, let's do a quick round of introductions with a check-in question that I'm recycling from an earlier call that Kamyar was on. And I'm going to have him go first because I thought his answer was pretty entertaining. So your name, your organization, and what is your summer bucket list item that you never seem to get around to? And then Kamyar, after you introduce yourself, please popcorn it to the next person.
Great. I'm Kamyar in Cheyenne at University of Northern Iowa, and the one that I never get to is washing the windows on our house. And I'll send it to Laura Merrick.
Hi, I'm a retired faculty member from agronomy in Iowa State University. I'm now Master's of Public Health grad student at the University of Iowa, and I couldn't think fast enough, so I'll just echo what Kamyar says, but it's not just something I didn't deal with this summer. It's been many years. Darren Thompson.
Yeah, so Darren Thompson, Associate Director at the Center for Health Effects of Environmental Contamination, or CHEEC, at the University of Iowa.
And where did Darren go?
Darren,
We'll have you pass it.
Yeah, you've got to.
Oh, I got to pass it.
Someone else.
Let's go to Victor.
Hi, everyone. I'm Victor Sapine. I'm a PhD student at the University of Iowa in the Department of Epidemiology. And I guess my bucket list item that I've always wanted to do was go to Glacier National Park. And I'm finally going to make that go through. I'm leaving on Friday. So I'll finally get that bucket list item off. Let's go to Dana.
Dana Copen, research hydrologist, U.S. Geological Survey. I guess my bucket list is actually finishing painting my house. It seemed like every year I get part of it done, but never get the whole thing completed. And pass it on to Greg.
Great. I'm Greg Lefevre. I'm a faculty member in IHR in Civil and Environmental Engineering in the University of Iowa. And it's hard. I have two very small little kids, and it's hard to think about bucket list because they were both vomiting on me yesterday. So that's my life update.
Oh no, we'll have you pass it to somebody, Greg.
How about Emily, because she's right next to me on this waffle.
Hi all, my name is Emily Dvorak. I work at the UNI CEEE with Audrey and Kamyar, and I manage the Good Neighbor Iowa program. And my bucket list item would probably be going on a bike camping trip. I went on one a few years back, and I've always wanted to do one each year since, and it just hasn't happened since that first year. So trying to make that happen again.
Nice. You want to pass it?
I'll pass it to Aaron.
Hi, I'm Aaron Lehman. I farm here in central Iowa, and I served as the president of the Iowa Farmers Union. My bucket list is to get to a Twins game every summer up in Twin Cities. I won't be doing that, I don't think, but I am going to get to the minor league game on Tuesday at the Field of Dreams before I won't get to the major league game with Rob's beloved Reds playing the Cubs, but I will get to the minor league game on Tuesday night. So that should be fun. So, and of course I have to pass it off to Rob.
Wow. The Reds are doing poorly, by the way, so whatever. My name is Rob Fox. I am communications manager for Pesticide Action Network, and I am also a farmer of a small scale diversified farm near Tripola, Iowa, known as the Genuine Fox Farm, if you all care. My bucket list thing that never seems to get done is I always want to get to August 1st and not have fields or crops I'm looking at in my diversified vegetable gardens that I'm like, crap, we didn't do that one right this year. Hasn't happened a single year yet, and it'll probably never happen because we just grow too many of them. And I will pass it. Who's next to me on the screen? Sarah, you're next to me on the screen.
Good morning, everyone. I'm Sarah Nizzi. I work for the Xerces Society as a Farm Bill pollinator conservation planner and NRCS partner biologist. And I'm not a real big bucket list person, but I'm always just trying to either bike or kayak or fish more every summer. And sometimes that happens and sometimes it doesn't. And I will pass it off to Bill.
Okay, I'm Bill Simpkins. I'm in year 2 of being a retired groundwater professor from Iowa State University. Did research and still doing research on water quality and groundwater, especially in the city of Ames, with all their issues at the moment. Claire is smiling, I can tell. So my bucket list, yeah, I'm not a big bucket list person. I just had a friend that hiked the Appalachian Trail. That's not on my bucket list anymore. But my bucket list is smaller. I just need to clean off my workshop desk in my garage of all the garden utensils and various other things that are sitting there because it's getting hard to find anything there. So that's on my bucket list. And hopefully that'll come in the next week or so. Okay, who is left? Oh, Penny and Jessica.
Now, can people hear me? Okay, yeah, we have 2 different ways of muting, so trying to figure out the combination of that. My name is Jessica Butters. I am the pollinator conservation specialist at Prairie Rivers of Iowa. And one thing that I've been wanting to do is go on a solo camping trip. However, I never seem to make time for it, and I never know exactly where to go for that. I'm always worried about safety going solo camping. So that's something I've always wanted to do. And if anybody has tips, let me know.
So I'm Penny Brown Huber. I'm the executive director at Prairie Rivers. And on my bucket list is I've sort of dabbled in a couple foreign languages. And I was just thinking about the other day, I'd like to become more fluent in a second language. So I'm going to hopefully get working on that. Oh, is there anyone else that needs to, Emily, maybe?
We got George, Roz, Claire. So maybe I saw George's hand up, so we'll go George, Claire, and then Roz.
Hi there. I'm George Hubble, OB-GYN, practicing in Missouri, of all places, but I'm on the PESU, the Pediatric Environmental Health Specialty Unit, for this 4-state region that includes Iowa. So that's why I'm here. My bucket list every year, for 55 years, I've been off on this week, which makes my garden quite drought ridden. and I just haven't come up with the way to get it all to survive. Only the strong things survive. And so anybody has any ideas on watering well away, let me know. Thank you.
All right, Claire.
Hi, Claire Ruby, DNR, PFAS sampling as usual. Let's see. I have a bucket behind me. Can you see my bucket? No. I want to go back to Greece. That's my bucket list. I haven't been for like 25 years. And I don't know if I'll ever get there, but I'm going to buy a moped and pretend, because I found a moped this week. So that's my bucket list.
Amazing. Roz, you want to round us out? I think that's everybody.
Okay, my mouse wasn't cooperating. Good morning, I'm Roz Lehman and I'm with the Iowa Organic Association. And I'm a bit like Victor. I have 2 children as well. So everything revolves around them, which has been all baseball all the time. So we've just wrapped that and we're actually getting a chance to just kind of do nothing. With that, I would have loved to sit by a pool, finish a book before school starts this year. I'm halfway through one, but haven't quite finished it. So fingers crossed on that one.
I love the variety of bucket list items from.
Oh, and Rob, by the way, the Cubs aren't doing that great either. So it could be a good matchup. So.
Okay, thanks everybody. Thanks for indulging me. For the record, my bucket list item is kind of a plot twist like Victor's, because I think it's actually going to happen. It's happening in a few hours. I'm going to go on vacation in a little while with my siblings for our annual SIB trip. Okay, so we are going to move on to the bulk of the meat of our call today, which is talking about neonics. So I'm just going to toss it right on over to Dana, who is going to give us the intro to the topic, some of the USGS effects research, stream exposures, and he will pass it over to Darren, who will then pass it on over to Greg.
Okay, can you see my screen?
It's perfect.
Awesome. So yeah, what I'm going to talk about today, a lot of it you probably already know, but I thought it'd be a good refresher as we transition to the newer stuff that Darren and Greg will be talking about. So really, agriculture is just it's ever evolving, particularly in terms of insecticides. It's changing insecticides for a variety of reasons, whether it's new compounds on the market, changing pest and resistance issues and so forth, and even changing in application techniques. And really, I'll get into the seed treatment aspect, which I think is really important in terms of neonics. But again, that's really how things are really evolved from way back in the 40s using DDT through the 90s and OPs and pyrethroids and up until the last decade or so with the neonic insecticides. So really the neonics have really very quickly become the most heavily used insecticide class in both the US and globally. A lot of the early effects research has been on bees and the colony collapse disorder issue, but there's growing evidence of broader effects to a number of both aquatic and terrestrial organisms, and increasing evidence of human exposure, which Darren was going to get into a bit later. So really, why are neonics popular? It's because they have a broad spectrum of insect control and really are widely used both on home and gardens and obviously with agriculture use, particularly with the seed treatment aspect. They're potent at very low doses, they're neurotoxic insecticides. They provide long-term control. In general, they're thought to be less toxic to vertebrates. And the important thing with the neonics is they are active systemically, which means the active ingredient is actually drawn up into the plant, and that's how it basically then prevents pests from attacking the plants of issue. But that's also can cause, that property can actually cause some collateral damage, unintended collateral damage to beneficial insects as well. So really the seed treatment aspect, which I noted, is just really exploded over the last decade. If you look at the USGS use that's been compiled, you can see that dramatic increase, but started in 2015, for whatever reason, now seed treatment use is not considered, I guess, a typical use. So you really have to be cautious of this use didn't drop off is just now the way the national county level use data was being assembled. The seed treatment aspect actually fell off the map, which is extremely unfortunate. Really, essentially now all the corn and a growing amount of soybeans are now planted in Iowa using treated seeds, which includes neonics and a range of fungicides. And from the industry perspective, they feel this really allows for target application. It's kind of precision agriculture, which this is my personal soapbox, maybe not a view of the USGS, but I think that is true on a field basis, where if you planted treated seeds versus broadcast spraying of a field, yes, you're using less pesticides, less insecticides via the treated seeds. But when you push the treated seeds to the point where every single kernel of corn, you got 14 million acres of corn in Iowa are now basically treated with an insecticide. I think that perspective is really, I think, lost, because I think we're now actually using more insecticides than we were historically, when we were only using insecticides to basically treat either active or pending infestation. So if you look at the map of neonic use, this is just one of them, clothianidin. You can see Iowa's right in the heart of the heavy use area. Again, this is the agricultural use, but and again, look at agricultural fate. This is, really focused on the agricultural side of things. You can see, because really neonics are both water soluble and persistent. They are going to be mobile. And when you only have between 2 and 20% of the neonic, again, with the treated seeds, is actually being uptake into the plant, that leaves a lot to potentially go elsewhere. And back in, say, around 2013, 2014, there was really a lack of information on what is happening. Where is neonics? Where are the neonics going after their agricultural use? And that's kind of where the research gap that my research team was helping fill. But again, in reality, it's not just the treated seeds. That's certainly an important aspect, but there's so many sources and pathways, and this just lists a few. There's municipal wastewater, so that's really a point source, being continually discharged into the bank. But it's, when you irrigate with wastewater, which it isn't as big deal in Iowa, but other parts of the country it is. So you have a points first source versus non-point source. You have municipal biosolids, which is taking place in Iowa. That is another non-point source for neonics. You had food processing plant wastewater, which really hasn't been thought of much until recently. We just published a paper a few months ago where we looked at about 23 plants across the country, including ethanol plants and meat processing plants and so forth, and found over 90 different pesticides coming out of these food processing plant wastewater, which included neonics in many of these plants. Livestock manure. Most people are thinking neonics is basically a crop issue. We're finding neonics and pesticides in livestock manure, and that gets applied to the landscape as well. So that's another non-point source, in landfill leaching. So again, the point is there's lots of other sources than kind of the more standard planting of treated seeds and land application to crops that we typically think about. So we did a study, and this was a few years ago. Basically, this is as part of our chemical mixture study, where we looked at hundreds and hundreds, I think about between 600 and 700 organic compounds in streams. We did a paper that focused specifically on the neonic aspect of this stream study. And basically, we found this is not just a Midwestern issue. We found neonics in over half the streams that we looked at. The big players were, in this particular study, were imidacloprid, clothianidin, and thiamethoxam, where clothianidin and thiamethoxam were related to, positively related to row crop production in the basin. But imidacloprid, on the other hand, was actually positive related to the amount of urban activity and urban land use in the basin. Focusing on now Iowa, again, not surprisingly, because Iowa's right of the heart of that heavy use map that I showed previously. We looked at 9 sites and sampled them multiple times across the growing season, and really found neonics in all 9 sites, including the Mississippi and Missouri River. So our 2 big border rivers still had neonics detected. All sites, multiple samples, almost ubiquitous, certainly in say the growing kind of planting season, almost ubiquitous. Just only one sample didn't have a neonic detected. So again, we're now approaching 80% of the samples had at least one neonic, with now clothianidin being the big driver for neonics, which I think is a number of reasons. First, of these three, it has the longest SOA half-life. It's heavily used. It's most exclusively used on seed treatment, but is also actually a degradation product of thiamethoxam. So it's probably a combination of all those factors why we found clothianidin so frequently detected in streams. We looked at one site in particular. We looked at basically frequently sampled Old Man's Creek just outside of Iowa City. Because of its close proximity, I was out there sampling every time it rained, whether that was a weekend, 3 in the morning, Easter Sunday. I was out there collecting samples at roughly, as best I could, kind of a peak flow. If it hadn't rained for a couple weeks, I would then just collect a sample, about every 2 weeks, and then more frequently whenever it rained. This kind of solid gray portion is roughly the planting season for this particular year. You can really see that you get this kind of classic spring flush that we've documented for atrazine, since the 80s. Herbicides, we know every growing season, every planting season, we get a flush of herbicides. But this is really the first time we've documented this classic spring flush phenomena with an insecticide. And it is the first time it's been documented that's taking place with seed treatments. So again, this is really a important aspect where maybe not surprising, but with now being an insecticide, which rule of thumbs, insecticides are generally more toxic than say herbicides, and that treated seeds is still causing environmental exposures to the point where are they problems? And that's what we're working on now. And actually just want to kind of wrap up. This is going to be a story for another day, but I just want to put a range of things that the Food IST, which the Food Integrated Science Team that Laura Hubbard and I are co-leading. We have a number of studies that we're really looking at, how exposure to neonics are affecting both aquatic and terrestrial organisms. Certainly the monarch butterfly, which coincidentally has just been listed as an endangered species. We've got a study on native pollinators that we did across Iowa that we're working on getting the papers written now and assembling this very complex set of results. Got terrestrial species with birds, earthworms, and then a number of things that actually are a part of the Alt-N, the Mead, Nebraska site that we actually just completed a food web sampling a couple weeks ago, where they really did intensive sampling of the food web associated with that ethanol plant, where there's astronomical levels of neonics are being input into the environment. And actually it's not related to Iowa, but we actually are also looking at other species as well, and how that exposure to pesticides and neonics may be affecting their population levels. So with that, I don't know if we just want to jump to Darren, or if we want to pause and take a quick question. I'll leave that up to you, Audrey.
Yeah, wow, thank you so much, Dan. This is, I certainly have a lot of questions. We do have a little bit of time. Our agendas they're typically pretty flexible. A couple of questions came up in the chat. I think we can spend maybe 3 to 5 minutes fielding some of the quick questions, but I may save some of the bigger ones for after Darren and Greg go. So George has a good question about actually fungicides in here, since I think you mentioned that neonics are typically applied in concert with a couple fungicides. Maybe we can revisit that one later. Laura had what I think could be a quick question for you, Dana. Do you think there's any way to petition to get neonic seed treatments added back for the annual assessment for USGS?
I haven't dug into that. I don't know if that's possible, because I think what we do is we purchase that use data, which then we process and assemble and then share. So unless the people we purchase from start changing things, I don't know how easy that is, but that's very, very unfortunate. And it's unfortunate on a number of levels. I think EPA, my understanding is they really don't even consider seed treatment kind of a real use. So there's a lot of kind of things that kind of loopholes that I think are unfortunate regarding neonics. But I will check into that, but I'm not quite sure if we'll able to get back to what we did previously and actually provide a truer data set on neonic use that includes seed treatment.
Yeah, and Rob just chimed in with that the EPA does not consider seed treatments. So it sounds like you're right there. I wonder if there's something that, oh, Darren, you go ahead. You unmuted. I didn't know if you had something to weigh in on.
Oh, no, I'm
just getting ready. Okay.
Getting ready. And
yeah. Sorry. So really quickly, I think that I can address this one really fast. George had asked a question about, given that soybeans are only treated with one third of soybeans are treated with neonics, is there significant yield increases with the application? I wish we had Jorgen from PFI on here. They looked at this.
And I think the third is probably an underestimated now. That slide, I pulled it from a slide deck I had. I would guess that's much higher now, maybe even above 50%. But what I've seen, and I can pull up a paper, is actually there was no yield increase, at least for the paper I saw, from planting with treated soybeans, because basically the biggest pest, at least in Iowa for soybeans, is the soybean aphid. And that generally doesn't take place till late July, early August. And basically what the protection you're getting from planting that seed is just too far away. So that by the time the soybean aphids are a problem, you're not getting any protection from a treated seed. Now, corn may be a different story. I haven't seen papers on corn to see if you get actually an improvement in yields with corn. Maybe somebody else knows. So maybe corn rootworm or armyworm, maybe there is some protection, but soybeans, I have not seen any. So, my personal opinion is I still think neonics have a useful purpose in the toolbox. The problem is when you're prophylactically using it, I think we're, potentially mismanaging neonics and using way more than we need, just kind of insurance protection.
Mm-hmm. Yeah. Okay, a lot to think about here. I know we'll circle back and pull on some of these threads at the end, but I hope that I've got Darren's slides up. Give me a thumbs up if you all can see that. Right, thanks, excellent. And we'll turn it over to you, Darren.
Okay, thank you. So I think I've presented here previously regarding some of the groundwater results. So I'll just kind of briefly summarize that. My interest is more in the human exposure and ultimately looking at kind of health effects related to that exposure. So I'll give an update on some of the research that is kind of hot off the press and hopefully will be published shortly. Next slide. So this is just kind of a summary of all the groundwater research that I've done previously. So I did a few studies, one looking at public water system wells across the state, then looking at wells, particularly in northeastern Iowa. And what we found is that clothianidine, imidacloprid, and thiamethoxam were frequently detected not only in groundwater, but also in the finished drinking water samples. And this was often detected in the summer and in the winter, which suggests that in particular, clothianidine is present year-round and may represent kind of a potential source of chronic exposure for people drinking water from wells or even from certain public water systems where they have vulnerable wells. In addition, environmental conditions such as pH, light's another factor that tends to break down neonics. So neonics being in the wells, not going to be exposed to kind of conditions that may break it down as readily as on the surface. And so this may actually drive some of the persistence. So clothianidine, for instance, is a neonic in and of itself. It's also a degradative of thiamethoxam. And there have been some studies indicating that it's may stick around as long as 16 years. So if it's being regularly used, the potential for it to be there for a long time are, I'd say, quite high. We also documented that there was an increased risk of contamination in wells that were closer to fields where pesticides were being planted and where treated seeds were being planted. And when I compared private wells, specifically vulnerable wells, to all of the public wells, we found that concentrations were much higher for clothianidine and total neonics in those private wells compared to public water systems. Now, when I compared only vulnerable wells from both sets, there wasn't as much of a difference, but that just, especially if you're living in somewhere like northeastern Iowa, where you have a lot of karst, kind of the right geological conditions for surface water contamination making it into the groundwater, the chances that you're going to be exposed to neonicotinoids is pretty significant. In addition, we looked at treatment. So we looked at treatment, home treatment and private wells, and then municipal systems. And what we found is that while concentrations may be reduced a little bit, it's not completely removing neonicotinoids in most cases. For the tests I've run in water, we look for 7 analytes. And so these are all the parent compounds. We didn't look for metabolites or degradants. So acetamidoprid, clothianidine, dinotefuran, imidacloprid, sulfoxaflor, thiacloprid, and thiamethoxam. Next slide. And so the next couple slides are just kind of some general graphs to show some of the trends we observe. So more often than not, shallower wells tended to have more frequent detection. So here, wells that were no more than 20 meters in depth, detection was 83%, whereas the deeper you got, so greater than 120 meters, only 7%. And this was for the public well samples, which came from systems from all across the state versus specifically focusing on Northeast Iowa. Next slide. And then detection frequency by land use, also the same set of wells that were tested from the previous slide. So again, clothianidine was very commonly found with row crops, and then forest and grassland seemed to be, half the samples had a detection, half didn't. And then developed samples, we also, not much difference in terms of imidacloprid detection, but significantly lower detection frequency for clothianidine versus row crop and forest grassland. Next slide. We also looked at detection frequency by season, so summer, winter. And again, generally, clothianidine was more frequently detected in the winter months, but still common in the summer. And some of this could be, again, that it's thiamethoxams being planted in spring, it's degrading over time. And so by the time we tested in winter, we're looking at clothianidine. But generally speaking, we had higher detection frequencies for all 3 of the neonics during the winter months. We didn't detect acetamidoprid, dinotefuran, or thiacloprid, or sulfoxaflor. Hold on a second. I have an 11-year-old who is. Yeah, go on. So no matter how many times you tell them that you're in a meeting and presenting in one ear and out the other. So Greg, that's what you got to look forward to in a few years. Next slide, please. So this kind of brings us to more of the latest results. And this was a study with Agricultural Health Study, and specifically they had a cohort of kind of a subset of members. So the Ag Health Study, I think, has around 60,000 members, pesticide applicators and their spouses in Iowa. And so we looked at a very small subset of 47 in northeastern Iowa. And with this, what we were looking for is kind of a worst case exposure. So we were looking at people who in the prior 12 months had used neonicotinoids, used private well waters or primary source of drinking water, and then we're drawing that water from a vulnerable source. So specifically either a alluvial well or a vulnerable bedrock aquifer. And here, what we did is we collected samples at the wellhead, and then out of the kitchen tap or out of the tap where they're getting their water for consumption throughout the day. And then we paired that with urine samples that were collected at 3 times. So in the evening, typically before bed, middle of the night, if they woke up to use the restroom in the middle of the night, and then first morning void. And so this was kind of done to see if we can associate what we were finding in the water with what we were finding in the urine. With the water samples, we looked at the same parent neonics that we looked at previously. With the urine samples, our intention was to look at the same 7, but we had some methodological method challenges with getting our analysis right. So we partnered with a lab at NYU who had done a number of studies all over the world, and they looked for 14 different analytes. So the same 7 that we looked for, plus a couple additional neonics, and then 4 metabolites of the 7 that we were looking for. These were collected from December 2018 to February 2019. Next slide. So this is kind of the geographic region in Iowa that we were focused on. Again, part of the state where we know that there's high vulnerability. So we would expect that we would see exposure within this population. Next slide. And looking at the water samples, it's fairly consistent with what we were finding previously. So again, clothianidine was typically in both the well and the tap water sample. We did find, I believe, 1 sample with dinotefuran in an outdoor sample, but not in the indoor sample. And then imidacloprid and thiamethoxam were also fairly consistent. I believe there are less than 10 households of the 47 where they were treating the water. And in general, with the exception of, I think, 1 household, the home treatment might have reduced concentrations marginally, but not significantly. We were still detecting it. And I think the 1 household we didn't find it. I think they had a pretty kind of unique setup where they were essentially, I think it was the water was being distilled and then sent through the house. So I think the expectation, you're not going to find much there aside from even the neonics. Max concentration again was clothianidine, 571 nanograms per liter outdoor, 141 nanograms per liter indoor. 13% of the households had at least 2 neonics detected per sample. Karst geology was associated with higher concentrations and more frequent detections as well. Next slide. So the biomonitoring sample, overall, we had 115 samples. So 47 first morning void, 24 middle of the night, 44 evening before. And so again, 14 analytes tested by NYU. So acetamidoprid, clothianidine, dinotefuran, imidacloprid, sulfoxyphor, thiacloprid, thiamethoxam. And then we had 3 other neonic, 2 other neonics and another neonic-like compound. So flunicamide, imidacloprid, which is a neonic that is produced outside of the US, and to my knowledge has no approved registered use in the US. And then nitinpurim, which to my knowledge is only approved for veterinary use. So if you go to like Walmart and you look at the flea and tick treatments, you'll see this as a very common ingredient in many of those. And then 4 metabolites, so 6-chloronicotinic acid, which is a common metabolite for a number of the neonics, acetamidoprid, desmethyl. And so there have been a number of biomonitoring studies that have been published kind of across the world. And this is typically one of the most common analytes detected in those. So acetamidoprid breaks down pretty quickly, and it may be one reason that we don't typically see it in environmental samples. But in the body, it's typically kind of excreted as this metabolite. thiaclopiridamide, and then indesmethyl thiamethoxam. So what we found was that on average, we were detecting 10 different analytes per sample for the urine samples, with a maximum concentration of 33.2 micrograms per gram, adjusted for creatine. So typically what we have here is concentrations that obviously the kidneys are concentrating it, but we're looking at levels that were significantly higher than what we were finding in the water. So roughly hundreds to thousands of times higher than what we were finding in some of the water samples. And so what we think is happening is that the water exposure through water represents a very, very small portion of that exposure. Typically, neonics are metabolized or passed through and excreted fairly quickly, some of them within 24 hours, some of them within 3 days. So likely what we're seeing here is fairly recent exposure. So this isn't going to be necessarily something where they planted in the spring and we're seeing the exposure later that winter. This is likely something they've been exposed to within the past several days. The two kind of individual analytes with the highest concentration per an individual sample were again acetamidoprid, indesmethyl, and then imidacloprid, which again is very interesting because it's not registered for use. And so the source of it is at this point is just kind of conjecture. It's likely that it's not being applied on the farms or being used typically for veterinary treatments. What is likely happening is it's coming through imported food and individuals are being exposed in that way. But again, we didn't look for some of these in water. So that's something that I'd like to do is to look for some of these additional analytes in some water samples to see if maybe they're present there. That might help to explain some of that. But I think food and other sources are probably explaining more of this. Next slide. And so what we found is very, very frequent detection. So this is kind of an average across the 3 samples. Acetamidoprid wasn't very common. But again, this is fairly typical and consistent with a lot of the publications out there. Again, what's happening is it's being converted to that metabolite. And as you can see, the NDMA was found in on average 83% of the samples. Whereas clothianidine, nitinpyrim, thiamethoxam, thiaclopiridamide were found in 100% of the samples. Imidacloprid was found in all but 1 sample. And then the desmethyl thiamethoxam was found in most of the samples as well. And so I actually had spoken to NYU about that. And I mean, they've tested samples from Asia, from all over the US, from other parts of the world. And what they essentially were saying that in terms of kind of the broad exposure and the frequency and detection, this is not something they typically seen. So essentially, even here in this population where we expected to see clothianidine exposure, probably some imidacloprid and thiamethoxam, here we're finding a lot of different exposures that can't readily be explained by the water sampling that had been done. So again, I mean, the denaturatin of the studies that I've done, I found it in 1 sample, and here it's in 70% of the urine samples that we tested. Now this could be, kind of a worst case. Again, these are people who are dealing with these things on a day-to-day basis. And so it may be that these results aren't generalizable to kind of the overall population. But, the fact that these are finding these concentrations at levels significantly higher than what we're finding in water, and that they're being exposed to so many different compounds that are probably going to act and behave very similarly, given kind of their mode of action, is something that kind of raised some eyebrows. And I'm have some studies that are kind of in the works where I'm hoping to look at exposure across more of a diverse population and then do a follow-up study to compare kind of exposure across Iowa, and then with people in Iowa and then people in North Carolina. Next slide. And then another thing, what I did is kind of looked at those exposure and trying to look at associations, correlations between water intake. And what we didn't find was that typically concentrations in tap water, I should say, were not correlated with the urinary concentrations. And this could be that, I mean, it was so frequently detected in the urine that it may be that we're not seeing here because there's not enough, I guess, difference in the concentrations that are being detected. A study with a larger population with diverse types of exposures may find something, although the indes-methyl acetamidoprid was positively correlated with the number of neonics detected in the tap water. So the more neonics that were detected, the higher chance that you're going to detect the indes-methyl acetamidoprid. Also, we did a survey looking at kind of farming practices. So these are self-reported pesticide application uses over the past 12 months. The urinary concentrations were also not associated with what was being reported in terms of seed treatment use and pesticide application. There was a positive correlation between tap water intake and frequency of detection, and an inverse correlation with bottled water intake. So people who drank bottled water typically had fewer detections for the neonicotinoids. So this suggests that there may be something there, but further investigation needs to be done, including looking for some of these other metabolites and compounds that we hadn't previously looked for. So generally speaking, again, I think, and again, consistent with other studies, is the concentrations in water are likely a small percentage of kind of the total exposure to neonics. We're likely just getting it from food, potentially occupational, dermal exposure, other things. But right now, there's probably more questions than answers in terms of that. Next slide. We also looked at exposure and diet. So each participant completed a 24-hour dietary recall. So basically everything they drank and ate 24 hours before I did a home visit, 24 hours before that first morning voids sample that they would have collected. And what we found is coffee intake was correlated with several neonicotinoids, including total neonic concentrations and the number of neonic detections. People who are consuming more than 4 cups of coffee a day had up to 10 times higher median concentrations of neonics compared to people who consumed less. And then as part of this survey, the daily dietary caffeine was estimated, and concentrations in milligrams of this caffeine were also correlated with, again, acetamidoprid, nitinpirim, thiacloprid, 6-chlorotinic nicotinic acid, and thiaclopiridamide. And then we also found associations with total fruit intake, which includes juice, intake of citrus, melons, and other fruits, with 3 neonics, sulfoxyphor, and a metabolite. And then we were somewhat limited on smoking history, as there were only 8 people out of the 47 who ever reported smoking, and only a handful of them were active smokers. But based upon those 8 people who reported smoking history, there did appear to be some correlation with exposure, like flunitramide and imidacloprid, as well as some other ones. But again, that's fairly limited. I think we'd probably need a larger population of smokers and non-smokers to really kind of test the accuracy of that finding. Next slide. So general conclusion is that in urine, neonics are widespread. Also fairly common in indoor and outdoor water samples. Exposure at this point is likely coming from a multiple different sources: water, diet, other occupational exposure. One thing that has been suggested is house dust. There's not been any studies looking at exposure through house dust. There are a few studies that have kind of documented transport of neonics through dust in fields. But that, I mean, that's just a couple of studies. So, I mean, these are things that I'm hoping to look at in the future. And then, of course, there are a couple of neonics that we're not exactly sure what the source is. So specifically the imidaclothis, which may come from imported food, but there may be other sources such as smoking. And then the nitinpirum, which again is probably coming from exposure to veterinary sources. But again, we didn't look for that in water, so it could be that it's in the water that people are drinking. But likely, if it is in the water, it's probably coming from these veterinary products that are being applied for flea and ticks and other pests. Again, observed exposure is likely very recent, just because of how these are being broken down by the body or how they pass through. And the assessment maybe worst case scenario. And I'm working on more studies that will hopefully kind of give a kind of a better picture of what kind of a typical Iowan's exposure is, regardless of profession or use exposure. Next slide. So finally, Cheek had been working to set up a lab, I think, for the past year, and just recently we finally got our instrument and everything working. So we're currently working on a method that will include many of the 14 analytes plus a number of other compounds, including pesticides that are commonly co-applied, so fungicides, nematicides, herbicides. So typically seed treatment will have a combination of those in addition to synergists and adjuvants and things like that. And so I'm really interested in looking at kind of this total exposure to what's coming off of seed treatment, because we do have kind of some very preliminary evidence that indicates that some of these other things were also in the water, but it was based upon a subset of 15 households that we sampled using a non-target analysis, which essentially is just kind of looking at mass for thousands of different potential compounds. And there were several things that we found in most of those households that, again, are typically in these seed treatments. And so what that broader exposure looks like is something that I'm hoping to define. In addition, now that I kind of have some of the exposure data, we'll be looking to start some studies looking more retrospectively at kind of potential health effects in cohorts within Iowa. So that's the final slide, and I think we'll hand it over to Greg unless there are questions that
Darren, that was amazing. Did you see the collective heart attack we all had when you talked about coffee consumption? I have a lot of questions. I haven't been able to take a look at the chat while sharing the screen, so I'm gonna sort of catalog questions as they come in. I'm thinking maybe we'll circle back to all questions after Greg's.
I will say that if you're drinking tea, you're probably not any better off. It's just these people didn't drink tea. And studies from overseas also show that tea intake is typically associated with this. In Japan, they've actually even associated it with like finger trimmers and things like that. So, yeah.
Yeah, neonics in Japan are a thing, like a separate thing entirely, right? We should set aside a little bit of time talking about that too. But Greg, I'm going to pass it over to you. And while you're getting your slides up, I just want to give a big hello to Mark Smith, who hopped on, I think, a little bit later. He is with our IDOL's Pesticide Bureau, so really glad you could be here, Mark.
Thank you.
Okay, over to you, Greg. Thanks. Oh, but you're muted.
There we go. Do you see the title slide that?
Yes.
Okay.
You're looking
Sometimes
good.
it flips back and forth because I've got a laptop over on the other side. So awesome. Well, thanks so much. I'm excited to be talking with this group, and I think this is a good stacking pairing of everything that we're kind of talking about here today in terms of different exposure routes and everything. And so I'm going to be focusing primarily on the work that I am, and my team, and this has been done extensively also with Dave Swartney in our department, as well as a whole bunch of grad students and stuff like that too. And we're going to be focusing mostly on sort of the engineered angle of drinking water and wastewater. So I'm not going to go into neonics and what they are, except to go in and to say that a little bit more about their chemical properties from the perspective of how it influences water treatment and why we see so many of them, right? So first, a little, dissecting their name, right? They're called neonicotinoids because they're based chemically off of nicotine. They target the same nicotinoic acetylcholine receptors as nicotine does, but they are neo, and so as far as they're new, and they confer much greater insect or invertebrate rather target specificity, because the fundamental structures of those receptors are very different between vertebrates and invertebrates. And so one of the way, or the way that target specificity is conferred is through some sort of highly electronegative group. So for neonics, it's either a nitro group, like is most common in the big three neonics, or sometimes it's also a cyano group. So which I think is like, for example, like thiacloprid, it has a cyano group on it. Either way, it does the same thing. Basically what it does is it makes it so it preferentially binds to the insect acetylcholine receptor and is basically repelled by the mammalian one. So I want you to keep that in mind, because we'll circle back to that and why that is important for the for water treatment, right? And you can also see these values here. This is kind of a fancy chemical term, but what it is the smaller the number, the more the compound wants to be in water. So like a lot of typical, like Dana mentioned, some of these legacy pesticides like DDT or something. This is on order of magnitude scale, sort of like the Richter scale, right? So DDT would be like 4, right? So that would be 4 or 5, orders of magnitude higher in terms of how that one does not want to be in water. Some of these, like thiamethoxam, is negative. So it really, really wants to be in water. So what that means is that, this is a study from Michelle Hladek and Dana showing that these are very frequently detected in our surface waters, and that is because they're very water soluble. So most of our drinking water treatment infrastructure is designed specifically to do 2 things: remove particles and pathogens, right? And pathogens are treated as particles, right? They're little bacteria, and we can go in and stick them together, remove them, and kill them, right? Well, things that are highly water soluble are not really designed to be removed. And so there's a lot of these more emerging trace organic contaminants like these are not, we wouldn't expect them to be removed in drinking water. And so based on this and their high frequency, we started working on looking at, well, would they be? So it seemed like a somewhat of a simple question, but important. And so we undertook a study. So this was actually in 2016 that we actually did this. We looked at neonicotinoids in the University of Iowa drinking water, finished drinking water, right? And so this was just like, post the planting season, so June, July, and we just looked for the big 3 compounds, and we found them in every sample. This was not terribly surprising. We also, this is the Iowa River flow. It seemed to maybe be related somewhat to the flow. It's a little bit hard to tell because we're downstream from a dam. So like the flow is anthropogenically altered, right? So it's not totally that way. But I mean, these are relatively low levels-ish, right? But still fairly high by most standards and stuff like that. And so, but this is the finished drinking water. This is the concentration of the tap water. When we looked across the university drinking water system, this is a conventional treatment train, right? So it goes from the source water to all these different unit operations. But again, it is going in and it's designed explicitly to remove particles and pathogens, right? So sediments, they go in it and, they're trying to kill those things and everything, because I mean, those are acute public health hazards, right? Those, we don't want E. coli in our water and all that kind of good stuff. But basically across the treatment train, we don't see any significant removal. There is actually removal of thimethoxam in one particular unit operation, which if you're really interested in that one thing, I'm happy to fill you in on that, why that happens. And it is a real thing. So we published that in 2017, and it kind of made a big splash in terms of like, got on the cover of the journal article, and my grad student and I were interviewed in the BBC World, and which appeared in apparently Buffalo, New York at like 1 a.m. On the radio as well. But it was, and also in the Washington Post and stuff like that. It was the first time people had ever found these in finished drinking water. And, the first question people would always ask is, are you drinking the water still? And it's, I oftentimes would demur on that type of thing or something like that. But then the second, and I'm also not a toxicologist, right? So putting these in the perspective of what that means is sometimes a little bit wishy-washy. But we also, at the time that this came out, had potentially, my biggest concern also is, remember first slide that I talked to you about, that the insect specificity is conferred through that, basically that one chemical functional group. And that's done on purpose. And so these are sometimes called target specific pesticides. And that's a really good thing, right? And what happens is that, the fundamental structure of the binding receptor and stuff like that, it goes in and it binds preferentially to the insect receptor. That's good. And it's designed to actually inflict less broader or collateral damage to ecosystems. And neonics are much less harmful to people and fish than a lot of other legacy pesticides that they replace. So that's a good thing. We can talk about, how that has perhaps indulged over usage. And I would also argue that things like, I have a friend who's a agronomic entomologist, and he said, we already see insect resistant crops, and or insect resistance in here, like, overusage has led to this problem and stuff. So if you want to keep this as a tool, don't use it all the time. But my biggest concern from a human health and a drinking water exposure perspective is that you can undergo some very small chemical alteration, right? Remember, there's a highly electronegative group over here. And if it goes in from being highly electronegative to one, right, if we were to remove that one nitro group here, it can actually flip the binding specificity such that it's actually much more likely to bind, which would mean greater toxicity to mammals, vertebrates, than the parent compound. And so this leads to really our central challenge of neonics. And generally, this is not exclusive to this. Other like fipronil is another target specific thing, right? You can have this where products are more toxic than the parents due to one small change. And really what we're interested in here is how that, that one specific functional group, which is called the pharmacophore, which think of the word pharmacy or pharmaceutical, right? It's some sort of chemical functional group that imparts biological activity, right? If you have some sort of transformation by bacteria, how would that affect water treatment? How would that potentially impact potential new products that are formed, like daughter products? like how would that also change the exposure spectrum? And so one of the things that we did was to go in and to look at several of these different products. Imidacloprid is, because it's the oldest, it has the most commercially available products available. And so one of the things that we did was we looked at to try and look at what would happen during chlorination, right? Water's always chlorinated to prevent, bacteria and viruses and stuff like that growing in there. So we were looking at disinfection byproducts, or DBPs. So we looked at imidacloprid, and we also know that microbial processes can go in and form desnitro or imidacloprid urea. We also went and basically did the world's simplest experiments, which is basically just taking some water and adding some bleach to it, right? Which is what chlorination is in a controlled manner. But then we used really advanced chemical analytical techniques to go and identify new products that no one had ever seen before. And so these are all things that can form in the water as a result of the chlorination. And we don't know anything about their toxicity or anything like that. A lot of these products are probably rather unstable. But one of the things that is a general rule of thumb, quote unquote, in environmental chemistry is that the more halogens, which include things like chlorine, on an organic compound, the worse that it usually is in terms of toxicity and bioaccumulation. So one of the things that we did, and what's neat about Iowa City, is that the university has its own drinking water system, which is a great place for our living laboratory. But the city of Iowa City also has its separate drinking water treatment system. And so one of the things that we did was we looked at the most common the common neonics in the university and the city system. And this was just a simple, in the source water versus the finished water. So just in and out, not breaking down what's going on inside here. And you can see, basically they have very similar source waters because they're both coming from the same river. The city uses some of the water gets filtered through the riverbank first, so it gets diluted a little bit, but very similar source waters. And essentially there's nothing coming out of the city plant. What's different about this here? Basically, very similar unit operations, except there's one difference, and which is that they have these large GAC, or granular activated carbon filters. And these were originally put in because a long time ago, apparently long before I was here, the Iowa City water tasted like fish all the time and was nasty. And so people put this in when they built the new plant. And it's fairly common as a way of controlling so-called taste and odor compounds, which is really what drives consumer complaints about water quality. You can't taste neonics in your water, but you can taste, wow, this smells like fish and nastiness. And so people put in these, the city of St. Paul has these big giant ones and stuff like that because they have taste and odor issues. And one of the things that we were really surprised about was that, remember how we said these are really water soluble? Most activated carbon is good for removing things that are not water soluble. So we were really surprised by this result. And so here's Craig, he's one of the managers, and here's some students. This is the city of Iowa City's water treatment plant. And we are seeing that is effective. We isolate just that one unit operation. We see that's very effective at removing them, but it was very surprising because they are so water soluble and very polar, right? And so one of the things that we did was we did some really pretty fundamental mechanistic studies. And one of the take homes that we found was that we could see, basically we're looking at how these things top out, and we see that activated carbon, first up, is actually quite effective at removing neonics. We dove into why, but one of the things that's a take home is that the parent compound associated with this electronegative functional group is much more effective, and it loses some of that efficacy when it changes the charge distribution due to transformation at that one group. So it's much more reactive towards chlorine, and it's also less likely to be sorbed on there. So we've also been doing some work, so advanced topic warning, right? Where we go in and we combine our experimental work with really advanced, using idealized surfaces like these fancy functionalized carbon nanotubes, and then computational chemistry, where we can then go in and combine these things into predictions to be able to predict the removal with activated carbon for compounds where there's no commercial standards yet. So this is a really powerful new tool that we just published earlier this year. So the other end of the water spectrum is looking at treated wastewater. So this is somewhat, we started this out at more of an ecological perspective. This is actually the same picture that Dana had earlier. This is a study site that we share, is right here in Coralville, North Liberty. And one of the things that we did was we, over the course of a year, looked at neonicotinoid or concentrations in the wastewater and in the downstream level. And to our surprise, we just kind of added this on to another study we were doing. And to our surprise, that some of the time in the stream, due to the wastewater, particularly from things like imidacloprid, was definitely driven by the wastewater itself, as opposed to chlorothianine tended to be somewhat maybe more baseline in the upstream levels. This stream actually kind of follows a rural to urban gradient. And so there was some chlorothianine in the upstream, but the imidacloprid is almost entirely driven by the wastewater. This is treated wastewater, right? So it's making its way through. And many of the samples are above the chronic invertebrate toxicity levels. Some of them are above the acute levels in here. So this is, we actually did some, our actual study that we were looking at was pharmaceuticals in here, which is what would be expected in wastewater. What we found was that actually from a risk exposure perspective to invertebrates, and is that it's actually driven by the pesticides which are in there, which makes sense, right? Because pharmaceuticals are designed to help people and pesticides are designed to kill things. So one of the things that was also perhaps an interesting thing that kind of circles back to something that Darren did was this is a very back of the envelope, right? But we looked at the levels of munics that were in the water, and we looked at what would be estimated in, this is a wastewater system. It's fairly new infrastructure. North Liberty is the second or first fastest growing city in Iowa. So it's built out quickly. Things are new infrastructure. It's not combined stormwater, wastewater systems or something like that. And looked at registered uses for these products in Iowa, and looked at, for example, USDA has allowable food residues and what people eat, assuming that are there, what are in flea and tick prevention, which is based on some studies in Europe and in California. This is actually a major driver of indoor pesticide usage. And one thing I always tell people is that, remember, if it's coming off of a cat or a dog and it's getting into the wastewater, cats don't shower, right? Last time I checked. And that means that it's getting from your cat to the drain. So you are probably being exposed in that process too. And one of the things that was really interesting too is that there's many multifamily dwellings in here, and it's become kind of a standard practice to do monthly pest control in many multifamily dwellings. And so people come in on a monthly basis and go in and just spray the baseboards of everything. And so particularly for highly sensitive groups like small children that like to go around on floors and lick things, that is a potential important exposure route too. So really the conclusions here from this work are, we know that they are in drinking water. We know that the neonics can form novel disinfection byproducts, of which we don't actually know the toxicity, some of which are probably fairly unstable and we know that they can actually revert back. So it's probably not a huge exposure risk, but we don't know what that is right now. We know that sorption or chemical sticking is driven by that one chemical group or that pharmacophore, chemical functional group that's there. And it's different between there. But the good thing is that activated carbon is actually a really pretty cheap way of doing water treatment. On a per basis for a municipal, they can get that fair. When I say cheap, it's relatively, right? It's much cheaper than something like reverse osmosis, which would work fairly well, or things like ozone, which would, blast everything to pieces, right? Obviously, communities that are much more economically disadvantaged wouldn't be able to afford something like this. But this is the same type of stuff that's in home Brita filters, or oftentimes home like basement filters or something. So they actually work pretty well for doing this unexpectedly. And we dove into the chemistry of why, but it is that's actually a pretty good solution for a lot of things in terms of end group exposure. So I want to also end with one thing that we found that was kind of a neat little solutions thing from, outside the engineering realm, is that we did one study where we looked at imidacloprid and thiacloprid, and we're really interested in kind of like a little bit of natural system treatment. One thing that's kind of curious, and we're following up, and we actually just published another paper. This came out yesterday with a group in Canada, is that duckweed are known to, which are, everybody's probably seen this if you've ever seen a farm pond, right? They're the world's smallest flowering plant, but they also have unique microbial systems associated with their roots that grow on them. And if you go in and take the duckweed and you just have the plants, or take the microbes off of them independently, neither of them can degrade the neonics, but together, and we don't know why, but they're able to successfully biodegrade at least these two neonics, and not form the toxic byproducts, which is oftentimes the case when you just have soil bacteria that are in there. So I will leave with that, because that's at least kind of a neat little hopeful note, is that there's something kind of cool with that too.
Yeah, that's helpful. Thank you for leaving us there, Greg. And just thank you for another wonderful presentation in our sort of barrage of amazing presentations today. We're going to jump right into Q&A. Hopefully you saw I put in the chat, shoot your updates and plugs to me so I can put them directly into our follow-up email, or you can put them directly into our notes document. I think we're going to skip that part of our call just so that we can extend the discussion and Q&A a little bit. George had a really good question about what's the best filtration system, which you just thoroughly answered, Greg, so thanks for that. But George also had a really good question about sort of looking at whether or not the concentration of neonics since you first detected them in treated drinking water in 2017. Have you kept an eye on that? Has the level of neonics increased at all? Has it stayed the same? Do we know the answer to that question?
One thing that's kind of an interesting thing about the Iowa or the university system, which is where we've had a lot more high levels and stuff like that, is that the university, because they have basically a very basic water treatment system, they actually they were getting violations for trihalomethanes.
Mm-hmm.
And which is a regulated disinfection byproduct. And in order to help ameliorate that, they added in a powder activated carbon system that they run only at different times of the year when there's high levels of organic matter in the river. But doing that, it's not as effective as the city's granular activated carbon system, but it does work to help lower levels substantially. And I think what's kind of cool about that is that because the treatment system was forced to go in and address a regulated pollutant, they also, because neonics are not regulated in a drinking water system, right? So by doing that, they was also incidental removal of a lot of other things.
Mm-hmm.
One thing that we worked with, we went and did some corings with the city GAC filters when they would normally go and they would exchange them, I think every 5 or 7 years or something. And so when they were about to do that, they let us take some cores right there. And what's cool is that we found, high levels that were in the cores, and that means that it was working, right? It was there. And we worked with the USGS pesticide lab, and they measured over 52, I think, different pesticides that were in the cores. And obviously we weren't measuring those in the water, but that just goes to show that, you can do things like address a regulated disinfection byproduct or taste and odor by doing this, and it helps remove all kinds of other things that are there too. So I mean, GAC is a known technology that's good for kind of a catch-all of a lot of things.
Yeah, doing what it's supposed to do.
We didn't expect it to, which is what was surprising and fortunate too, because it's not expected to work on these types of pollutants.
Right. John Fagan, thanks for being here, John. Also put in the chat, activated carbon is also really effective in pulling glyphosate and its byproduct AMPA out of water systems too, which goes to what you were saying about the pesticides, Greg. Darren, I had a really quick question for you about the biomonitoring study. Did any of those 47 households have pets that would have contributed to
We
what
did
you were
look at
finding?
that. I don't think there was enough to really draw any conclusions because there was only a handful who reported using basically treatments on their pets in the past year. Yeah, I mean, I think we did find something, but the numbers were just so small that it's something we decided not to report, because it's like if 2 people report using it, you find something.
Right.
I mean, it's not something that would have been publishable. I'm hoping again, so intending to look at like almost 400 people across Iowa, North Carolina from the same cohort, see if we find something similar.
Yeah.
So a larger number, we might be able to draw better conclusions.
More to be discovered here for sure. Just like all of the questions that there are around the synergistic mess of all the chemicals that are applied in mixtures together on this. And so I'm glad that that's a topic of interest for you researchers here. There was a really important question from Kamyar that I don't know if we sort of danced around it a little bit, but what are the do we know anything about the human health impacts of chronic neonic exposure, of acute neonic exposure?
There's not a lot published out there. There's more than when I started my dissertation. So it's kind of, when I started, nobody was doing biomonitoring, and then everybody was doing biomonitoring. And now there's gradually studies that are kind of more mechanistic that are coming out. Like I think I've seen a couple of studies where they found some associations with like insulin and glucose homeostasis, but it's fairly mixed results. I think there was a study out of Cincinnati that linked neonic exposure to lower testosterone levels. But a lot of these studies are, smaller cohorts than what I was dealing with, 19, 20 people. So again, in Japan, there's been some studies looking at kind of acute exposure, consumption of tea, and finding kind of nerve tremors. But again, these are a lot of kind of not as in-depth as probably is needed to draw any firm conclusions. I mean, what I do know about toxicology, what we find in the water is well below what would be considered kind of an acute or chronic threshold. I mean, significantly lower. I mean, I think less than 1% of what they believe that you can drink. But again, those things are constantly being updated. And again, those are looking at individual neonics. They're not looking at what the multitude of neonics and potentially other pesticides of other families that have similar kind of functions that you're being exposed to. But I mean, at this point, the concentrations appear to be within safe levels, but I suspect within the next 5, 10, 15 years, we'll have a clear picture of whether they're actually safe, and actually kind of concerns me given what I saw in this population.
Right.
And the neonics that aren't even used that are being exposed to at levels higher than anything we're finding in what is being used on a day-to-day basis. So.
Right. And Dana, I see that you're unmuted, so I want to pass it over to you.
Yeah, just really quickly, we've talked about, potentially get exposures through drinking water, consumption of via our diet, whether it's fruits and vegetables, but we haven't really talked about air. Is this another going to be, it's the cumulative of all the potential pathways, and we're breathing, pesticides routinely, and either Greg or Darren would have mentioned household dust. So there's all kinds of pathways. So, any one compartment is just going to be a partial of what our total exposure is.
Right. And John threw in the chat here, the difference between the EPA threshold and what you'd find in the recent literature is really, really quite big. The things that you mentioned, Darren, about maybe some touching into the human health realm of this, being those metabolic disorders and low testosterone, that kind of gets into some of the other chronic low dose exposure stuff that we see with other types of pesticides too. So anytime in environmental health, how to start teasing apart these variables is just going to make your job a lot more harder as a researcher. I want to circle back to a question that George had way at the beginning, linking in fungicides to some of this. Dana, you had mentioned that like neonics are often applied in seed treatments with maybe, I think it was 1 to 5 fungicides. And I think, and it has been at the top of the call now, so I don't remember the exact nuance of the question, George, but is there an issue with sort of plant pathogen resistance with application of these fungicides in the face of climate change? Do we know anything about this that any of you
those
feel
are great
like?
questions. I don't think we know that yet, but certainly we do know that, as has been mentioned, it is going to be a mixture issue. So neonics as a class, you can kind of sum them all up and feel safe that they're kind of have similar modes of action. But when we know there's exposure to fungicides and herbicides and the transformation products of all the above, that's where it just starts snowballing quickly to get beyond what we really understand in terms of what are both to the environment and to us personally in terms of exposures to these broad mixtures of not just pesticides, but pesticides and PFAS and pesticides and everything else we're getting exposed to. That's where it gets extremely challenging.
Right. Darren, I see you're unmuted.
And I think that's why you don't see a lot of studies looking at mixture, because it's so complicated. I mean, even aside from the active ingredients, I mean, there's a synergist called piperonal butoxide, which, I mean, you go to Walmart and you look at some of the pest treatments, and it's there. And my understanding is the way it works is with the insect, it essentially makes them more vulnerable, so that the active ingredient has a better, I guess, efficacy. And then the non-target samples we found, I mean, it was one of the most common things we were finding in those 15 samples. The non-target analysis doesn't tell you concentration, it just tells you what's present. And so that's, these things may not be toxic, but what if you have something like this or other synergists in there that are actually, making your immune system, your body more vulnerable to what would typically be kind of non-toxic effects. Again, I think I've seen one study looking at piperonal butoxide with another pesticide, but just not enough people doing the investigations on some of these things.
They oftentimes are ignored from a regulatory or even from a research perspective because of the fact that they're not listed as the active ingredient. They're considered a NERF because it's not the goal of that specific product to do that specific thing. It's, they might be completely intended to be biologically active, but it has other things. And so, yeah, it's a very challenging thing. I wanted to just add on one quick thing about like how much we've learned in what Darren's point about how much things have changed. Also, like, we might have put this out in terms of drinking water exposure and stuff like that, but there's been so many studies worldwide now in terms of demonstrating that these things are in treated drinking water and drinking water sources, that it's just, that's why it was like kind of a dumb luck thing to show this. We were not surprised at all as far as those things. So that's why it was like important, but not necessarily surprising. Some of the other things, as far as like the fact that activate carbon works, that was really surprising. And there's a lot of cool science behind that too. So, but I think, there's definitely been a lot of new knowledge that has come out from this. And I think we can also, think about what these things mean in a big picture context.
Right. George, I see that you're unmuted, so I'm going to let you wrap us up.
Not to wrap up, just unfortunately not the question, and then I did have kind of an overview, but one, all these chemicals as a class have one mechanism of action, and that is the postsynaptic acetylcholinesterase junction. And as such, just lost my earphone, but as such, I would think that, most of the side effects, therefore, if one was to, again, peer into what they're doing, would be a neurologic side effect. But unfortunately, with toxicology, it just takes gargantuan amounts of chemicals or toxins to have an impact on the neurologic system, as opposed to, other organ systems, i.e., the reproductive health system and endocrine modifiers. So, versus neurologic systems are really, really difficult to certainly see, short-term or, or long-term effects of these neonicotinoids. But anyway, that was just my 2 cents.
No, it's really, really good to have that physician perspective, George.
But and then finally, there's kind of a parallel in medicine, and I don't know if you guys all have PBMs, but they're pharmacy benefit managers, and they're kind of this useless teat on a board. Just drive up the cost, and they are outside the regulation of the ACA, and therefore companies have just been pushing them to a great degree, and are a profit center for a lot of these pharmaceutical companies. Well, the parallel here would be, these particular, if we don't see any benefit on beans, why are all the, they have some great salesmen out there, some great marketing people to be endangering us all. And I would think that this would be, an easy target for legislation, or some introspection into truly, what they're doing to us.
Thank you, George. Thank you so much. I mentioned in the chat, there is so much left here to discuss. And I did, of course, like I typically do, a bad job of budgeting how much excitement and time we would need to really dig into this. So I want us to keep this going. One of the questions that I had for Dana, one of the bigger questions, having to go back to the data collection for neonics and seed treatments, is how do we as a group have this conversation with USGS, or the folks that you are purchasing the data from, USGS is purchasing the data from, so that we can know what's out there and how to be talking about it in an accurate way. So basically, that's what I was going to talk a little bit about in my brief update to the group. That I think that there is a real potential for getting in, besides doing the really important co-learning and sharing, but to also move a little bit more into action. Like, how can we be, doing work in Iowa that complements the research, the really amazing research that's being done in the state? All TBD. You have indulged 6 extra minutes of your time for this. I really want to thank so much Dana, Darren, and Greg for joining us and for sharing outstanding presentations. Hopefully we can have you all back when we want to continue this discussion, which I know we really want to, and to include Dana and Sarah's especially perspective on the pollinator piece. Also, Penny and Jessica, I'm sure you have a really big piece of discussion to sort of share with us about the pollinator and other ecological side of the neonic discussion. We should move outside of humans, because that's a really important part of the web of public health here as well. Okay, yeah, we are atrophying our group because we've gone over the allotted time. So we will pick this up next quarter, and I'll send a follow-up email about all of this stuff when I get back from vacation. Thank you all so much for your time. It is not taken for granted. We'll see you soon. Bye.
Thank you.
Thanks for having us.
Bye.
Thanks, Audrey. Thanks, Audrey.
Thank
That was
you.
great.